Gordon Burghart's lab at the real UT, as Rita has instructed me to say, University of Tennessee. And then she went on to, well, she did her undergrad at Cal, then her PhD at UT, postdoc in the Wainwright lab, Peter Wainwright's lab at UC Davis, before starting her faculty position at UC Santa Cruz. I met Rita when I was postdocing in Peter's lab as well, and I'm sure Rita could tell you some.
I can tell you some stories.
There's a Barracuda incident that, depending on the perspective, was either me heroically saving the data that we collected, or from other perspectives, leaving a postdoc to be eaten by the Barracuda. The real truth is lost to the mysteries of time. We'll never know. Rita is a splendid anatomist and functional morphologist, evolutionary morphologist. When I was a PhD student, I was thinking about doing measuring muscle activity during striking in snakes. I talked to David Kundal, one of the foremost authorities on snake functional morphology. He said, "No, Mike, do not do that." But you have EMG data on constriction in snakes, don't you? Yeah. So Rita has been able to do some things that are very difficult and amazing, and reveal some really fascinating, provide fascinating insights into the relationship between form and function in organismal design. She's been working a lot on eels and other elongate vertebrates, which is what she'll talk to us about today. So take it away, Rita.
Okay. Well, thank you so much. I've had such a good time. So first thing I wanted to do, or I thought I would do, is I'd introduce you to my lab, which is known as Elver, a name for a juvenile eel. And so Elver has been growing over the last 4 years. I've got 4 graduate students. I've been fortunate enough to host 2 postdocs, and I've been working with over 20 undergraduate researchers, both in the field as well as in the lab. And I've been able to lure them into the lab with the help of our STEM diversity programs at UCSC as well as CalTeach. So those 2 programs have been very instrumental in me getting the critical mass I needed to help set up the lab. So the couple of questions that motivates the Elver Lab are, what are the factors that contribute to variation in individual performance? What are the factors that promote or constrain morphological diversity? And thirdly, how does body shape influence feeding mode? I like to ask these questions with eels, and as my mother might ask, Rita, why eels? Well, I think eels are really fascinating for a variety of reasons. First, they're elongate, and elongation really sits on the extreme in terms of the body shape continuum in all vertebrates. And I think from a physiological perspective, you can really learn a lot, as shown by other very well-known physiologists such as Knut Schmidt-Nielsen, that you can learn a lot by researching organisms that sit at the extremes. Secondly, because they do sit at the extremes, although all vertebrates are to a certain extent gate limited, it provides some really interesting challenges for feeding. So now what you have to do is cram food down a relatively small opening to sustain a large body whose mass is spread across a very long area. And then what this does is it also increases the surface area to volume ratio, which makes this body plan quite energetically expensive, which is why we mainly see it, with a few exceptions, in mostly ectothermic organisms. Also, which is also very interesting to me, is elongation is the ancestral body shape in fishes. Extant species, we calculated 20% of all extant fish species are elongate. And what's really cool is that elongation does not constrain body size. So you can have some really small elongate pipefish like Cyacunectes that have a commensal relationship with sea anemones, or you can have something as astounding as the oarfish, right, which is like 36 feet in total length. So, And that also enables me to meld my two interests, which is looking at body shape and feeding really, really big in the aquatic environment. Lastly, I have to say, eels just have this very interesting look to them, right? I mean, I feel like eels always look like they're kind of just told a joke and they're waiting for the punchline. But in all seriousness, I wanted to walk you through 3 vignettes. I'll probably be going very quickly, so if you have any questions on the methods, please do not hesitate to ask. So I'm going to be walking you through 3 projects: feeding mode spurs morphological evolution in eels, variable responses to durophagy, and then a project that is extremely new that I've been able to start up while at UCSC is looking at functional ecology of the California moray in the field. So let's start with feeding mode spurs morphological evolution in anguilliform fishes. So we feel like one of the main factors that might drive phenotypic diversification is the dependence upon, or the interaction of skeletal traits. So normally skeletal traits are very dependent upon one another to provide a specific function. One really great example of this is in suction feeding in the aquatic environment. So, you know, biting is a pervasive feeding strategy in the terrestrial environment, but it's not so much so in the aquatic environment. So this is an example of a tarpon that's using ram and suction to feed on this small piece of prey. I forgot I have to walk over here to do this. And so what you'll see is this very rapid behavior, right, where the fish is able to capture its prey item without even touching it. And what that depends upon is musculoskeletal elephants moving in a highly coordinated fashion very rapidly. And so how does a fish do this? Well, fish have the most highly connected skulls of all vertebrates. And when you look at the fish skull, it's hard not to notice that there are lots of skeletal elements, right, that combine to form the skull. And we can divide these skeletal elements into 3 different functional units. So we can look at the units comprising the oral jaws, the units comprising the hyobrachium, and then if you look at an anterior dorsal view of this radiograph of a snook, what you can see is the opercular flaring, so that water can flow through the fish's mouth, and that is comprised of a series of skeletal elements in functional unit 3. So elements work together across these functional units to provide a very rapid behavior such as suction. This is a really fun video that one of my former lab mates in the Wainwright lab, she was able to model suction feeding. And how we model feeding in fishes is using an expanding cone model so that the fish is expanding its skull anterior to posterior, and Kristen was able to capture this very nicely using some morphology from Centrarchid fishes and MATLAB to model this. So as you can see, the mouth moves anterior to posterior, and that's how that negative pressure is generated across the skull to induce a flow of water through the skull. Well, so although suction feeding is the most pervasive feeding mode in the aquatic environment, alternative feeding strategies such as biting behavior have evolved. And I hope that you noticed from this slide, there are very different skull morphologies in biting, because biting is not all the same. So there's different types of biting strategies. So we can contrast these 2 different feeding modes, suction feeding and biting. And what we find is that, hands down, suction feeding requires jaw abduction. You've got to open your jaws, and you've got to do it very rapidly in order to generate negative pressure. Also, the timing of suction feeding requires coordinated skeletal elements working across those functional units that I showed you before. And then lastly, if you look across all different suction feeding groups, what we see is that the behavior pattern is very phylogenetically conserved. So you always see this anterior-posterior movement across very disparate suction feeding groups. Now, if you contrast it with biting, the first thing you notice is you're not dealing with jaw abduction. You're dealing with jaw adduction. So an organism is slapping its jaws shut in various ways in order to catch its prey. So when I say in various ways, it means it can be fast, or it can be very forceful but slower, which leads to the second point. The timing of biting prey or capturing prey by biting is much more variable than suction feeding. Also, we can securely say that because the timing is variable, it's not requiring the same degree of coordination as suction feeding requires. And therefore, what we can say is that suction production is highly constrained. It's not just phylogenetically constrained, but it's functionally constrained because you have to have these skeletal elements that are working together across these three functional units to provide suction or to produce suction. That's not the case in biters. Biting kinematics are highly variable, which is why anguilliform fishes happen to be a wonderful model to study the evolution of feeding mode and how feeding mode affects diversification in the skull. So the exciting thing about anguilliform fishes is not only are they all elongate, but they're actually very ecologically diverse. And we find suction feeding lineages as well as biting lineages in this group of roughly 800 members. And so our hypothesis is that the origin of biting released constraints on skull diversification imposed by suction feeding. So once again, suction feeding is highly constrained, and the evolution of biting most presumably moved the dependence of the functional groups, allowing more diversification across the oral jaws, hyoid, and opercular series. So we have two predictions for this project. Is that one, biting lineages experience weaker evolutionary correlations among skull functional units compared to suction feeding. And secondly, weaker evolutionary correlations in biters is going to facilitate accelerated rates of diversification of individual functional units. So I could not do this without the help of two people, one being Mike Alfaro, who's a collaborator on this project, and he helped put together this unpublished anguilliform phylogeny that is comprised of 3 mitochondrial genes. And this was such an arduous task, I have to say, because what's hard is that Mike and I have to coordinate not just getting tissue, but getting tissue for specimens for which we actually can obtain morphological data. So there's a lot of coordination involved. In fact, when we go to museums, oftentimes, Mike would come back in an email saying, I can totally get these tissue. And then after doing a bunch of research, I'd come back saying, there's no way in heck I could possibly get the measurements for these species. So there was a lot of coordination to build this tree, and this tree is made up of about 124 species. So what we were able to get is morphology for 62 species of varying individuals for each species. And we collected data, as you can imagine, within the 3 functional units. So the oral jaws, the hyoid, and the opercular series. We size corrected these data using the geometric mean of head dimensions. And then we coded for feeding mode. So for many of these species, I was able to get feeding mode because I could bring them in the lab and I could see if they were biters or suction feeders. Others, some of the deep sea species, we have no idea what they were doing. We could rely on the literature for very few of them, but then for others, we actually had to figure out a way from the morphology, looking at fossil fishes as well as extant fish species that suction feed, what most likely makes a suction feeder. And then we coded based on those 4 characters, which I can talk to you about later. But they were different characters from what we used in our data set to avoid circularity. Lastly, we constructed ancestral feeding modes on our phylogeny using SIMAP. And this slide is specifically for Blair, so I'm so thrilled that she is here. So she could see, Blair, my fish are diverse. So similar to Blair's carnivorans, we see a lot of skull diversity in these fishes. And what I'd like to call to your attention is that you see a lot shorter jaws relative to overall skull length in suction feeders. You also see, I'm sorry if this is too small, but there are insets in each of these pictures, which shows the ventral side of the skull of suction feeders so that you can actually get an idea of how the hyoid apparatus has been modified for suction feeding. So first off, there are more elements in the hyoid apparatus in suction feeders compared to biters. There's a lot of reduced bony elements in the hyoid apparatus. And also the jaws are much longer in biters, and the teeth, of course, are a dead giveaway. You can tell biters from suction feeders just almost by looking at the teeth. So when we look at our ancestral reconstruction of feeding mode, what we see are a few different things. So biting is in black, and in gray you see suction feeding. And what I like to call your attention to first is that anguilliforms are the sister taxa to the Allopomorpha, which are comprised of some really fascinating and unusual fishes, such as tarpon, ladyfish, and bonefish. And what we see is that suction is the ancestral condition in the Allopomorpha. However, biting is the ancestral condition in the anguilliforms. And then what you see is that suction evolved independently multiple times in anguilliform fish. So this was a very neat surprise, which made anguilliform fishes, in a way, an even cooler model to look at, because usually what you see in most teleosclades is that suction is the ancestral condition.
Rita, that's
Yeah.
a parsimony reconstruction?
That is so what we see is we've got our Bayesian, this is the asterisk represents 90% confidence. So on the tree, that's right. But this is a likelihood reconstruction. Yes, thank you. So what we wanted to do, going back to those questions, is looking at correlated evolution across the different functional units. And so I was fortunate enough to work with David Collar, who I hosted as a postdoc in my lab for a little bit. And Dave is really, really wonderful at devising new methods to compare different groups. So what he was able to do, along with Liam Ravel, is come up with a matrix approach to look at the evolution of correlations. So the first thing that we did is to reduce the dimensionality of the data set, because I didn't tell you, but we had over 20 individual morphological measurements that we took, and that would be very computationally prohibitive. So what we did is we reduced the dimensionality by looking at a PCA of all the traits within each functional unit, and then taking PC1 from each functional unit. So, and again, I want to repeat that the data set were already size corrected. So PC1 is not an axis for size, that has been removed, but shape is still in there. So we used Dave and Liam's evolutionary correlation approach to measure how tightly integrated the functional units are between biters and suction feeders. So what I'm going to show you here is this intermodule morphospace, where each of the axes represent PC1 from each functional unit. So the oral jaws, the hyoid, and the opercular series. And what you can see here, biters are in the black triangles, suction feeders are in the white circles, is that they take up very different areas of intermodule morphospace. So biters have much longer jaws, they have much longer maxilla. The opercular series in suction feeders is much less diverse. Biters occupy a greater area on this axis, on the opercular series axis. And then for PC1, hyoids are very long in biters, but they're much shorter and more robust or wider in suction feeders. So what we can see here is that biters are actually occupying much more intermodule morphospace. Oops, excuse me. And so one of the things we wanted to ask based on this too is, how do the evolutionary correlations look across the different functional units? Are they different between biters and suction feeders? So what I'd like to walk you through here is this graph. We've got the functional units that are being compared, and this is from the evolutionary correlation matrix. We've got units 1 and 2, which are the jaws and the hyoid, units 1 and 3, which is the jaws and the opercular series, and units 2 and 3, which is the hyoid and the opercular series. And then on the y-axis, we have the evolutionary correlations. And what we see, again, biting in black, suction in white, is that we see high integration or high correlation, evolutionary correlation, between the jaws and the hyoid in both biters and suction feeders. So as the oral jaws are changing, so are the hyoid apparatus, so are skeletal elements in the hyoid apparatus. When you look at the jaws in the opercular series, what you see is that there's a tight correlation in suction feeders in the negative direction. However, there's nearly no correlation in biters. And if you look at units 2 and 3, the hyoid and the opercle, what you see is, again, very different patterns. You have biters, the correlation is not significantly different from 0, but you see a strong negative correlation in suction feeders. So what we can conclude from this is that, yes, the evolutionary correlations between biters and suction feeders are different when we look across the 3 functional units. All right, so I think what was really interesting with this result is then it sets up to look at the last question, which is, do biters and suction feeders differ in their rates of evolution in these different character traits? And, you know, my hypothesis, or what I predicted, was that yes, biters would be evolving at a much faster rate, even when you control for time. Well, what we find is really surprising. So I know the variance is very high, and that has to do with the fact that we looked at over 1,000 trees when we were looking at our evolutionary correlations and our rates. And what we find is that there's no difference in the skeletal elements across all these functional units. So when you control for time, biters and suction feeders are diversifying at roughly the same rate. There's no difference in rate. So what we can conclude in this project is that the transitions in feeding mode from biting to suction feeding, yes, have had a very strong role in diversifying the skull of anguilliform fishes. So there are shifts in the strength of evolutionary correlations across these different functional units. And biters do occupy a much greater area of intermodule morphospace. Suction feeders, as we anticipated, because they rely on all 3 functional units to produce that anterior-posterior wave of suction feeding, have much tighter evolutionary correlations across those 3 functional modules, whereas biters do not. And then the surprising conclusion is that we found no difference in the rate of diversification between biters and suction feeders. And so, you know, one of our hypotheses as to why this pattern is happening is that biters, again, are very diverse. they take up a wide area in that intermodular space. And so suction feeders, in order to evolve the correct characters to produce suction, they must have to pretty much evolve very quickly, because what you saw is that suction is the derived condition. So it would be really interesting to look at the flip side, where you have suction evolving first, or being the ancestral condition, and then biting repeatedly evolving to see if there then would be a difference in evolutionary rates. Okay, so now I'd like to talk about our second project, which is titled Variable Responses to Durophagy. So as you just saw, feeding mode can have a very strong effect on skull evolution. And I think one of the mechanisms for this strong effect is not just prey apprehension, but what the predator is actually feeding upon. And durophagy, the ability to consume or manipulate hard items in the oral jaws, is one dietary specialization that has converged repeatedly across vertebrates. So we see this pattern in sharks, in fishes, in lizards, and we see it many times in mammals. And so it's a very interesting dietary pattern to study convergence and try and understand what are the mechanisms that actually produce variation in the skull. So I'd argue that morays are a really great group to study convergence in durophagy, because we already know that skull traits conform very nicely to feeding habits. And also, there are genera of moray that we know are durophagous species. So it turns out that this genus that Echidna nebulosa belongs to is paraphyletic. So durophagy has evolved multiple times in the Muranidae. So the question we wanted to ask is, do the repeated morphological traits that we see in durophagous morays resemble a pattern of convergence in multivariate space? So essentially, I like to look at this question, or paraphrase this question as, how convergent is convergent? Like, how similar do you have to be termed or stamped, this is convergent evolution? And so what we did is we applied a definition by Tristan Staton at Bucknell University, who was looking at convergence in multivariate space for herbivorous lizards. And the way he defined convergence is morphological convergence is going to be supported, in this case, if my durophagous species occupy a region of morphospace that is distinct from their non-durophagous relatives, but that region of morphospace also has to be smaller than that's occupied by their non-durophagous relatives. So the first thing we did is we used a tree by Rees et al., and Josh Rees was a postdoc in my lab when we started this project. And we looked in the literature, trying our hardest to find diet data for all the species of morays that Josh had in his phylogeny. And what we were able to find is that in 70% of the cases where diet data were actually available, there's a pretty nice dichotomy when you're looking at diet. Morays that are termed durophagous, we defined as those that were consuming prey that comprised roughly 70% of their diet or higher. And then those that were not really were eating mostly softer but elusive prey. So we felt like the dietary category was really good, durophagous and non-durophagous. And for the sake of this talk, I'm just calling non-durophagous piscivorous. So this is a modified phylogeny from Rees et al. What Josh did here is he used SynMap to reconstruct dietary states, durophagy and non-durophagy on this phylogeny. And what we can see is that piscivory, based on this phylogeny, is the ancestral state. So Piscivory is in gray, and black is transitions to Durophagy. And we can see 10 independent transitions to Durophagy from a most recent piscivorous ancestor. So what we did is there were 46 species in that moray phylogeny, and unfortunately we had to prune them because I couldn't get all of the species for the morphological data set. But we were able to get 40, and we looked at across 176 individuals. We measured 9 skull variables. We measured actually many, many more, but these were the most meaningful measurements, of which adductor mass and tooth dimensions and outlever lengths of the lower jaw were included in this analysis. So we used a phylogenetically size-corrected PCA by Revel 2009 to look at how these morphological elements were dispersed in morphospace. And what we see is really interesting. So we've got our piscivores mostly on the upper right, and we've got our Durophagus morays in this region of morphospace. And so these are PCs. Sorry, I didn't mention that first. So PC axis 1, what we can say is that the traits that are driving differences in piscivores and Durophagus morays are the length of the maxilla, the mass of the adductor mandibulae muscle, and also the length of the supraoccipital, which is a skeletal element at the back of the skull that provides extra muscle attachment for the adductor. So as we would predict, Durophagus morays are doing some things that are separating them from piscivores, and these some things that they are doing are helping them to increase crushing forces. On PC2, it's pretty much all about the teeth. So Durophagus morays mostly have shorter teeth, and piscivores have much longer, skinnier teeth. But the interesting thing is if we relate back to Staten's definition of convergence, we see that easily Durophagus morays do set out in their own area of morphospace that's distinct from their most recent piscivorous ancestors. However, when we calculate the variance, we find that they have 2 times the variance, 2 times the morphological variance than piscivores do. So that's really perplexing because it only half meets the strict multivariate definition of convergence. So is this convergence? Well, it half meets it, right? But we got to go by the definition, and so no. But this is really unsatisfying because I would still argue that this is convergent. So we tried another tactic to examine morphological convergence. And what we did is we applied the Adams and Collier method, where you're looking at the evolutionary trajectories, so vectors from Durophagus lineages from their most recent piscivorous ancestor. And we're calculating the variance between those vector trajectories. And what we see here, the most recent piscivorous ancestor is in gray. Piscivores are in white triangles, and then black circles are Durophagus species. What we see is that even though the evolutionary trajectories or the vectors from the most recent piscivorous ancestor are going everywhere, when we calculate the trajectories for the Durophagus lineages and compare them to the trajectories of the piscivorous lineages, and the piscivorous lineage trajectories are not on here because it would just be really ridiculous to look at. What we see is that the variance in the trajectories for the Durophagus species is much less than piscivores. So that was validating, at least, and that enabled us to stamp, okay, this is an example of convergent evolution on this example of morays.
So before you leave that.
Yes.
Sorry.
No, it's good.
So you said the vectors, the arrows from the
Most recent.
piscivore ancestor to either the Duraphagous species, or I guess reconstruction of the.
Piscivorous, yes,
Yeah.
lineage, yeah.
But it's from Piscivore to
Duraphagous,
Duraphagous.
yes.
But that variance in the directionality or the size or something of those vectors is less than what?
It's less than the piscivorous trajectories.
So that's Piscivorous, so extant Piscivorous species.
Extant
back to
Pisc-
their
Most recent common
where
ancestor.
there wasn't a transition.
Exactly. Yeah, so transitions to Duraphagous species versus non-transitions, right? Because the ancestral state is piscivory. Yeah. Okay, so why is there more variation? Well, unfortunately, there's not a lot of time to go over all the different methods that we use to look at why there's more variation. But it's really fun to talk about when we think about really what could be causing, 2 times the variance in morphological traits. Well, first, it could be adaptive peaks, right? So we could be looking at the Duraphagous condition having multiple adaptive peaks. So not all Duraphagous prey are the same. So perhaps Duraphagous species are still converging in one area of morphospace that's distinct from piscivores, but they all have their own adaptive peaks. So we set a 2-peak model and a multiple peak model within Duraphagous lineages. And what we see is that only the 2-peak model was validated, so that there's only 2 peaks. There's 1 peak for piscivores, and there's 1 peak for the Duraphagous lineages. So we can say that it's not because of being multiple peaks in Duraphagy. Oops, excuse me. And then we looked at individual character responses to Duraphagy. So how are the different skeletal elements changing? Are they the same? And also lineage-specific responses to Duraphagy. So how are the lineages behaving in the suite of Duraphagous responses? So in terms of that first question that we asked, the multivariate peak, what I wanted to show you is that here you've got morays that are consuming just hard body prey, okay? So 100% of their diet requires crushing. And you can see this is just the Duraphagous morphospace. The piscivores have been removed. They're all over the place, okay? So that's another piece of evidence that shows us that there's not multiple adaptive peaks. Okay, again for Blair, here we've got lots of morphological diversity in Duraphagy. So, it's not surprising that they're taking up such a large area of morphospace. So this is an example of a piscivorous ancestor. And the most compelling traits that we measured were supraoccipital length, which I mentioned earlier, does provide a greater attachment point for the adductor mandibulae muscle. A vomerine tooth that usually is very long and skinny in piscivores. Maxilla length, the depth of the lower jaw, as well as the length of the lower jaw. And I hope you can see here that, many of these traits are changing, but not all in the same way. They're moving in the same direction, but that's how we get some variable responses. And if we look at a table of characters, for those specific characters that I showed you in the cleared and stained specimens in the previous slide, what we can say is, with a lot of confidence, there are 3 traits, 10 out of 10 times, and 10 times being the multiple evolutions to Duraphagy. These are changing in a significant direction. They're all getting shorter every time. These characters in yellow, what we see is it's marginal. It's not exactly significant. It's trending towards significance. Perhaps if we had a larger sample size, we had a larger tree. But in 9 out of 10 lineages, you see that these traits are changing. And then again, here, there are 3 traits that are changing in very different directions. So we've got traits that are changing in different directions that are contributing to the variance in morphology. Now, what's really exciting is when we look at the lineages, so we're actually putting all of these traits together, so this is a multivariate approach. What you see with each of these 10 lineages where durophagy has evolved is gray stands for traits that are changing in the opposite or unexpected direction. Yellow is traits that are changing in the expected direction, but they're not significant compared to the piscivorous ancestor. And then red are traits that are changing in the right direction. So we expect that these traits are getting smaller or taller or shorter to increase crushing performance. And they're significantly different in their direction of change to the most recent piscivorous ancestor. So what we can also conclude is that individual traits are changing in different ways, and the lineage-specific responses are very different as well. So what we can conclude is that, first of all, I wanted to bring your attention to this paper by Mahler et al. That came out in Science very recently. And their paper was exceptional convergence in the Anolis lizards. And it's very true. If you read this paper is a case of just striking convergence across these different islands in the Caribbean. You get very similar patterns in these adaptive radiations every time. So it's a really great classic model to look at convergent evolution from a macroevolutionary perspective. However, I think what we would argue is that when we're talking about convergence, it's not always exceptional, it's not always perfect. So what we see here in the case of morays is a case of imperfect convergence. So we see that there's a consequence of variation among traits in the degree to which they converge in the different lineages. And this variation definitely highlights that the relative strength of natural selection on durophagous feeding habits versus the lineage-specific responses. So in other words, natural selection acting on these traits for a durophagous diet versus contingency or evolutionary history that's driving these particular lineages. All right, so the last project that I'd like to talk with you about is a field project that I started about 2 years ago on Catalina Island. And the premise for this field research project is based on the idea that we know from functional morphology studies that moray eels have this really incredible adaptation for consuming large prey whole, and not only the aquatic environment, but morays can actually come up on land and feed, because as far as we know, they are the only species of fish that's not relying on suction for both prey capture as well as transport. So we know that morays can consume large prey. And in laboratory studies, what I've done, of course, is try to feed morays really big, like snakes, and just see how big of prey can they actually consume. And I'll tell you, they really can consume large prey. But the question is, what are they doing in the wild? Okay, so they have this mechanism, but are they really putting it to use in the wild? So this project is being completed, in progress with the help of my graduate student, Benjamin Higgins, who is out on Catalina working on trapping morays and looking at their diets. So this is a really great research project that we do every summer, and we hope to continue doing to collect long-term data on moray eels. So Catalina Island is one of the Channel Islands off the coast of California. And again, I think the reason morays are such an interesting group to look at is there's roughly 200 species. They're mostly tropical in their distribution, so the California moray is our only temperate species, and it's our only species off the coast of California. We know from studies in the tropics that moray eels definitely shape the future of community structure on coral reefs. So Mark Carr, who fortuitously is in my department in the 90s, was able to remove a bunch of morays from coral reefs in Hawaii, and he saw, compared to reefs where he did not remove the morays, that there's totally different fish communities that colonize or repopulate the reefs. So first of all, what he saw is that there's a lot of more juvenile fish on reefs that have morays removed. And also, you get a lot more diversity of fish in the night. So morays are known to be more nocturnal in Hawaii. A recent study on stable isotopes in the South Pacific just corroborated the idea that morays seem to be apex predators on many coral reef ecosystems. So that gave us more impetus to look at the California moray and what its diet is all about. So when we think about our kelp forest ecosystem off the coast of California, what we see is a diversity of fish species, and many of these fish species mainly come out in the daytime. However, the California moray is quite elusive. Its elongation enables you to hide in really tight crevices where you can escape from predation, or you can kind of have an ambush feeding strategy to feed on small prey that come into your crevice. We know very little about the California moray, so we know that it has the potential to be an apex consumer, as we've seen in tropical reef studies. We know very little about the abundance, very little about their range around Catalina, and there's only been one diet study that was published, and that was in the 1950s, and it was on a very small population of the California moray around Catalina Island. Also, what's really interesting to note, and I touched on this earlier, is that the reef or the kelp forest community is very different in the day compared to at night. So at night, a lot of these fish that are usually found in the water column come down and end up sleeping or end up very still around the rocky crevices. So there's very different profiles in the day as well as night. So our long-term research questions was to better understand the diet of Gymnothorax mordax, and also to understand whether or not this species has a particular preference for a habitat type around the Catalina Island area. And also, what is the distribution of the prey items or the potential prey items? And also, what are their movement patterns? So I'm going to be focusing on simply the diet and our methods to get diet. So we were able to find 4 really nice coves around the west side of Catalina Island where we do a lot of trapping and measuring and pit tagging. And so far, over the last 2 years, I've been able to bring out a really wonderful team of undergraduate researchers to help in this effort. And so what we do is we trap in the evenings, we pull traps early in the mornings, we take measurements. There are about 15 morphological measurements we take on all of our morays, including mass, and then we pit tag these morays. Now, for some of the morays for which we could get acoustic tags for, we pull them from the population, take them back to the lab, implant an acoustic tag so that hopefully in the future I'll be able to tell you more about the movement of the California moray. And then after we do all this stuff, we put them back into the water, and as far as we know, they do really well. So one of our, actually our only method for getting diet is by manual palpation. And it's really amazing that we can do this, but morays are snake-like predators. I mean, this is like the most non-invasive way you can get prey from a snake is by manually palping it. And essentially what you do is you massage the prey that you can see in the moray's stomach up and out of its mouth. And you actually can do this. So sometimes it requires that I take some needle drivers to do this, but other times, especially when you catch morays that are consuming octopus, it's really easy to push the prey out of their guts. So what we did in first pass is we used a linear selectivity index to estimate what the contribution of prey was in the diet of morays, where R here is a proportion of items in the gut, and then P is a proportion of items in the habitat. And what I should emphasize is Ben has been wonderful about assembling a separate diving team to go out and do visual senses in the different areas that we're trapping so that we can better understand what the prey composition looks like in the day as well as at night. So what we've done is we've actually trapped and tagged 585 individual morays in a 1,300 meter of coastline. And this year we were able to get 18% recaptures, so 106 individuals. And what we see from our different coves is that there's not exactly a significant difference in the amount of morays we're getting. What we are going to go back and we're going to analyze a size distribution to see if maybe size is different across the different areas. Our diet results reveal that really 33% of the diet of the California of the California moray is kelp bass, 26% are rock shrimp, 16% is octopus, and then 10% are rockfish. And then the rest are a bunch of invertebrates or unidentified fishes. So where do they fall out in the linear selectivity index? Well, what this index tells us is that morays are either avoiding these prey, they're either generalists, or they're highly selective for a particular type of prey. And what we see from the preliminary data that we have is that they fall into the generalist category. Now, what's been really fun is looking at the relationship of prey standard lengths that we can get and the different morphological dimensions that we've been capturing with the morays. And what we see is that there is a pretty strong positive relationship between standard length of prey and the vertical gape distance of a moray. So that's one of the morphological measurements that we've been taking. So hopefully in the future I will have some more exciting results to present on this field project, because we have a lot of different ideas. So first thing we do want to do is write an addendum to the California Fish and Wildlife to compare moray size in the marine protected areas versus out of the marine protected areas. I should say that where I'm working, there are so many marine protected areas. So that 1,300 meter of coastline that I'm working on is like the only non-marine protected area. So it'll be interesting to see if there is a size difference. We'd also like to extend the study out to different areas in Southern California. So I don't know if you guys know, but morays stop at around Point Conception. So they're found from Santa Barbara all the way down to Baja. And then the other thing that we're going to be looking at, I have an undergraduate who's looking at the dimensions of the teeth in morays to see how they compare to other piscivorous moray species. And we just completed a bite force project on Duraphagus morays. So we have our bite transducer, and what we'd love to get is population data on bite force in morays, especially how bite force changes across ontogeny. So with that, I'd like to end, and I'm sorry, hopefully we have time for questions. But thanks for your attention.
We have time for questions.
Yeah.
I take it from your graphs that you didn't obtain any larval morays.
Oh, no. In fact, So this is the big kicker with Catalina and Brad. I would really love to hear any insight you might have, either now or later. But so morays are not breeding in Catalina. So no one, as far as I know, who's been working on Catalina, for eons, like I ask everybody, there's been no Leptocephalus larvae found. So what we think is that the leptocephali come from Baja or around the Galapagos, waft up, settle in Catalina, and then Catalina is like the dead end.
No. You don't think there's a breeding population?
I mean, wouldn't we find things like Leptocephalus larvae? I mean, the smallest eel we found is this big.
35 years ago, Boyd Walker told me about this. He said, do not let a student study this because they will fail. They're not there.
Well, that's not this level.
Yeah, anything smaller than the adults that she's done.
That's fascinating. Wow.
So have you ever opened up females to, there's no possibility that there's direct development and they see.
No. I mean, I have found eggs, but I mean, no one has ever seen the leptocephali. I mean, we would anticipate that with like all the midwater studies going on there, that someone will report seeing a leptocephali.
Are they known? Do they find them other places? I mean, do they find for this species? Do they find them in?
They find them in San Diego. They find them
They find
south.
them, they do. Oh, okay. I just studied gene flow and
Yeah.
that'd be fun.
Yeah, Blair.
Yeah, really nice talk. I guess you are doing some performance work. So
Yes.
the natural thing is that big spread of Durophagus eels, are they all equally good at crushing what's happening in
Right. So that's what we are working on now. So, I think we are able to get about 7 of the Durophagus species that are out there. And, I'd predict that they're not all as good as the first one that we studied, Gymnomurena zebra. So it's going to be interesting to see, where they do fit on that performance landscape. It'd be cool.
And did you say something about, how long they've had to evolve in these directions? Is there any association between that?
Right, no, so that's an excellent question, and I did not get to that. So when we looked at the half-lives, apparently they're, let me try and remember. So with the evolutionary half-life, it looks as still, what we concluded is that there is no, hold on, let me try and get this right. It's a really good question. We did look at evolutionary half-life. But what we see is that it's not because they've had less time to evolve. That's exactly it, yeah. That's not the case. Yeah, Mike.
So seeing the first 2 studies right next to each other reinforced the idea, I think, that when you look at a multivariate morphospace and you're assessing patterns of convergence,
Yeah.
that convergence or lack of convergence reflects the rate of evolution.
Mhm.
It reflects the degree of correlation amongst the traits.
That's true too.
And
We don't.
time.
Right.
And so if you look at in your first plot with the suction feeders,
That's
like
good thinking.
Bristol,
Yeah.
which is really interesting to me
Yeah.
is that those guys, you show that there is no difference in the rate.
Right.
But if you look at the region that's occupied by the suction feeders, they've converged. Well,
That's
I
right.
didn't address convergence in that study, but they're in a much smaller region.
That's right.
And the pattern of correlation is actually strong in
Yeah.
those guys, right?
Right.
And then if you look at the durophages example, you have evolution when they go into durophage. You didn't look at the rate
No.
specifically in that.
No.
But I guess the correlations are also stronger for the durophages than the non-durophages, or
We
not?
didn't look at that.
Yeah, so I think, I don't know, I think there's something else there, like another way to add to thinking about, what is convergence, and also how do empirical patterns
Yeah,
inform, or what,
produce
like, what phase are we catching convergence, the process of convergence with
Yeah.
empirical data?
Yeah, so I feel like, that does touch on definitely Blair's question, but also I feel like you're trying to get me to say many to one mapping. Because, you can have multiple, where you have the
Yeah.
multiple lineages response to durophagy that is definitely, historical contingency is driving that. And that is really strong, but then you also have multiple ways in which you can increase crushing performance. And you've got variation within each of those traits too. So some traits are going to be more like plastic than others. Like the teeth are changing like crazy, and they're changing in the same direction every time. Whereas there's other traits such as the area of the ethnovomerine complex, so essentially the top of the head, which you would think, oh great, increasing a crushing surface area would be a good thing. But sometimes that decreases, sometimes that increases. Sometimes that is actually not different from the most recent piscivorous ancestor. So yeah, there's like lots of interesting things that are going on. And I think that's why that problem was such a challenge when we were trying to tease apart really what is convergence, and yeah, what can we like definitely say about these character traits.
Yeah, I think there's a lot more to explaining that your stuff is getting at. So.
Yeah, Brett.
So I hope I have it straight, but the 10 examples,
Of
10
durophagy,
case
yeah.
studies of durophagia. Okay, I think I have this right. So there, I think you showed you were talking about rates of diversification, or was that no?
That was in the other
that
project.
That was in the lighting.
Yeah.
Okay.
Yeah.
And that's rates of morphological.
That's right. That's rates of morphological diversification. Both of those
So
are.
what about rates of lineage diversification? I mean, so if I remember right from that phylogeny, there were one-offs where you only had a single species, but then there were also ones where there were clades of sort of 3 or 4 species. And
Right.
so, what about, when you make a shift,
Mhm.
does your rate of speciation or rate of species accumulation change? Is there, enough cases where you can look at that, so that when you make that shift, it changes your rate of speciation or your rate of species accumulation?
Okay, so I think our issue with that is that's an awesome question to look at, and I think Mike and I talked about looking at that question a long time ago. It's just that you would need at least 70% of the species for that clade, I think, to look properly at rates of speciation, and really we're looking at a quarter. So we really
You
need
just
to.
can't get 'em, or-
We did some pilot stuff with
Yeah.
that, and there's nothing jumps out, but there are
Where
a
we
lot of-
bootstrapped, right? But we still had
Yeah.
such a small sample
Yeah,
size.
there's a lot of issues, even though the phylogeny, there's a lot of work to be done on the phylogeny still. Even we had
And that was a purely mitochondrial phylogeny.
we have an updated version of that tree that we recently published.
He's talking
that-
about the moray phylogeny.
Oh, the mores, yeah. Oh, is that all? I don't know. Okay, so anyway, I'm just talking about like across anguilliforms. There are lots of taxonomic issues as well as phylogenetic issues that make it difficult to assess character-dependent diversification right now. So, but from the stuff where we've tried to do it, I haven't seen anything that's associated with these shifts from one to
Interesting.
another. But
Because it's a nice rich, there's a lot of shifts, and so that's
Yeah.
at least a good thing, right?
Yeah.
You've got a lot of replicates to look at.
Yeah. No, it would be wonderful. If we can get DNA from formalin-fixed specimens, then we are set.
You and the rest of the world.
Yeah.
All right, well, let's let Rita finish and get some lunch. But if you have further questions, you can catch her afterwards.